An ongoing project conducted at the University of Minnesota Wet Laboratory is mussel host suitability trials. Today we will be looking at such research for the Minnesota native and endangered mussel, the rock pocketbook. The rock pocketbook belongs to the Mollusca clade, a clade which includes well-known members such as the cephalopods. However, the species that we will be focusing on, Arcidens confragosus, is a freshwater mussel and a member of the bivalvia clade. For the life cycle of the rock pocketbook mussel, the female mussel broods eggs in her gills until they become larvae. In the fall, the mussel releases the larvae, also known as glaucidia. These glaucidia, only 1/3 of a millimeter long, fall to the river bottom and await unsuspecting fish hosts. Unlike many mussels, the rock pocketbook does not rely on attractive mantle displays to attract fish hosts. It simply releases and hopes for the best. Glaucidia that attach to fish's gills remain attached for around 2 weeks, while they metamorphose into juvenile mussels. Once juveniles, they drop to the river bottom to continue their life cycle, still only about 1/3 of a millimeter long. In search of rock pocketbooks to use for the host suitability trials, the research team spent multiple days snorkeling in the St. Croix River in known rock pocketbook mussel beds. None were found. Fortunately, the Minnesota Department of Natural Resources allowed the research team to use 2 of the 3 rock pocketbooks from their labs. Electrofishing was used over suitable mussel habitat in the St. Croix, both to attempt to collect naturally infested fishes and to collect a variety of larger fish species to use in trials. No infested fish were found. Also, to gather smaller fishes, seining was used both in the St. Croix River and in local streams. In the wet laboratory, glaucidia were flushed from a gravid female rock pocketbook. This was done by gently prying the mussel open, making a shallow cut along her gill sacs, and using water to flush out thousands of the glaucidia. These glaucidia were then placed in an aerated container to await infestation. Fishes to use for the trials were picked and placed into buckets. Glaucidia and an aeration hose were also placed into these buckets. This formed a glaucidia jacuzzi. Fishes were left in the glaucidia jacuzzi for 30 seconds, after which time they were removed and their gills were examined under a microscope to verify infestation. All fish species showed attached glaucidia. Fishes were then placed into species-specific aquaria. 3 times a week, the bottom of each tank was siphoned and run through a sieve. The siphonate, or the matter caught by the sieve, was then examined for both untransformed glaucidia and transformed juvenile mussels. If a fish species produces juvenile mussels, it is then considered a suitable host. While this study is still currently underway, previous rock pocketbook host suitability trials have shown a wide variety of suitable hosts. Why are these findings important? The rock pocketbook is currently listed as an endangered species. Like many mussel species, it fulfills a key role in the balance of many freshwater ecosystems. Because the rock pocketbook can use a wide variety of fish hosts to complete its life cycle, other factors must contribute to its rarity. Some evidence suggests that increased water pollution or sediment deposition may have caused the decline in numbers. Knowledge gained from the host suitability trials, combined with further research, will help us to better understand freshwater mollusks and preserve their role in the ecosystem.